Hello. So next we'll discuss about M phase. That is mitosis. So like if I just look at the overview, it was like G1, S, G2, then comes the M phase. In M phase here we have 4 stages: prophase, metaphase, anaphase, and then telophase. This all constitutes karyokinesis, and after that we have cytokinesis. This is the main idea. Okay, so now let's start with M phase. Now here because since it's mitosis, so number of chromosomes, they will not increase, they will remain the same. So number of chromosomes in the parent, okay, and in the progeny, they will remain the same. Progeny means offspring, child from okay, so chromosome will remain the same. That is why it is called equational division. Question is, why mitosis is called equational division? Because the number of chromosomes in the parent as well as in the progeny remains the same. That is why it is called equational division. Okay, so now mitosis, it has 4 stages. That is all probe, meta, ana, and telo. So all these together they constitute karyokinesis. Karyokinesis is basically what? Nuclear division or division of nucleus. So now coming to first phase, that is prophase. So this is the first stage of karyokinesis. Now here what happens? The chromosomal material condenses and it will form compact mitotic chromosomes. So here we have two, early and late prophase. So here prophase, now how it starts? Here we have initiation of, there is condensation of chromosomal material. Condensation of chromosomal material. So this chromosomal material, that will become untangled during the process of chromosome condensation. Or you can say during the process of chromatin condensation. So if I say condensation is like see, tangled, there are two terms, tangled and untangled. If I say like this, okay, this is in a cluster, that is tangled. If I say like this, this is untangled. Like in layman language, tangled means like which is in a group, which is not sorted out, which is in a haphazard way. So that is basic. Like you say, his hair are tangled. So that means that is very messy look. So tangling is like that, and untangled you can like very clear and sorted out. That is untangled. So condensation, chromatin condensation, like it happens like this. If I say untangled, or you can say condensation of chromosomes. That means in condensed form we can see as individual chromosomes. We can make out like this is a chromosome, but in tangled form, if there is no condensation, if they are in uncondensed form, we cannot see them as discrete individuals, or we cannot see them as individual chromosomes. Okay, then now there is a term centrosome. So centrosome which has undergone duplication during S phase. Now the centrosome will begin to move towards opposite poles of cell. I'll tell you what centrosome is. So centrosome, it will radiate out microtubules called esters. Okay, so here next is it in this prophase. Like if there is early prophase and one is late prophase. In early prophase, now this is basically this is nuclear membrane. Okay, so in early prophase, these are chromosomes are like this. But in late prophase, they become like this, then this. Okay, like we can see like in condensed form. So now here in prophase, they will also form mitotic apparatus. Now what happens in mitotic apparatus? Let's say this is a chromosome, and this is the centromere, and these are sister chromatids. Okay, this is the terminology of this. Then like here we have a kinetochore. So after centromere there is kinetochore. Okay, now from this kinetochore, what arises? Esters, like microtubules are there. These are microtubules, or you can say spindle fibers. Spindle fibers or microtubules. So basically these are made up of esters. Okay, now there is another term, that is centrosome. Centrosome, and then we have a term centriole. Now let's say if this is a cell, and here it is like this, and here it is like this. This is basically what this is, a centrosome. this centrosome, these little two, these, like, if I'm drawing like this, that means these two, this and this. This is basically centriole. For example, if I just to show you, this is a centrosome, okay, and these two small like this, these two small units, these are centrioles. Okay, so this centrosome, it consists of two centrioles which are placed at right angles. So these two are centrioles only. So here, like, mitotic apparatus in here, now there are two features here, chromosome. Now what happens in prophase? Chromosome condensation and formation of mitotic apparatus. This was all about prophase. And one more thing, no, like, cell organelles. If I talk about cell organelles, no Golgi apparatus, Golgi complexes, then endoplasmic reticulum, nucleolus and nuclear envelope, they are not formed. Golgi complex, endoplasmic reticulum, nucleolus, nuclear envelope, they are not formed at prophase because this is just the starting. Okay, so this was about it. Thank you.